Welcome to this presentation titled Impact of Porosity in Bioglass in Tissue Scaffold Obtained by Different Techniques. A 45S5 type bioglass is produced through a direct casting process. Using a platinum crucible, the mixture is subsequently heated to 1400 degrees centigrade for 2 hours. The melt is poured onto a copper plate and the subject to ball milling with ceramic balls. After reducing the particle size, a diameter of 0.020 millimeters is achieved, resulting in a bioglass with an average particle diameter size distribution of 3,100 nanometers approximately. Through scanning electron microscopy, particle size diversity is observed. Particles exhibit diameters under 1 micrometer. They present electrostatic attraction that induce their agglomeration, and a portion of them generate a surface roughness effect, where the largest bumps correspond to the larger particles, inducing significant surface roughness on the order of 2 micrometer maximum. When these particles are analyzed by atomic force microscopy, it is observed that they are formed by a series of sheets joined together to form an agglomerate with a maximum particle diameter of 1.5 micrometers. In a higher resolution analysis, it is observed that these particles are formed from a series of agglomerates with approximate diameters of 2 to 5 micrometers. In order to obtain smaller diameter particles, another technique has been used for the synthesis of the bioglass. For this purpose, the salt shell technique was employed. In this case, a bioglass containing calcium oxide and magnesium oxide molecules is synthesized. It should be noted that their ions act as network modifiers that induce the amorphous nature of the bioglass structure. This formulation of bioglass was synthesized using an acid-catalyzed such as nitric acid. Results from scanning electron microscopy show a particle size of 563 approximate nanometers. It is noticeable that the particle diameter is smaller than that obtained in the casting process, resulting in less raw surfaces. It has been estimated that these particle agglomerates generate smaller interparticle porosity than in the case of the bioglass obtained by casting. The type of particles obtained is a very diverse, since open pores, closed pores, not penetrating pores, bottle-shaped pores, and penetrating pores are found within them.
In order to obtain particles that do not agglomerate with each other, the synthesis of bioglass catalyzed by a base, such as ammonium hydroxide, was chosen. Scanning electron microscopy analysis show that it is possible to obtain silica calcium oxide phosphorus pentoxide bioglass nanoparticles with diameters greater than 20 nanometers, smaller than 100 nanometers, thus forming mesoporous particles. According to studies conducted by other research groups, these particles form an internal porosity with diameters smaller than 2 nanometers right from synthesis. In our case, the average diameter obtained was 79.918 approximate nanometers. An analysis of surface roughness shows that samples with a smoother surface tend to minimize the roughness between particles of the agglomerate. Thanks to his feature in particle agglomerates, it is possible to obtain hybrid organic-inorganic polyvinyl alcohol tissue scaffolds by electrospinning, thus enabling the insertion of particles with diameters under 60.350 nanometers approximate into fibers with a 100 nanometer diameter. A fourth experiment was conducted where an organic-inorganic polyvinyl alcohol bioglass hybrid with a silica calcium oxide phosphorus pentoxide composition was obtained. The compounds that form the bioglass, such as tetraethyl orthosilicate, 3-ethyl phosphate, and calcium chloride dihydrate, are added in situ to a flask where the dissolution of polyvinyl alcohol in water takes place. The process takes place at 73.5 degrees centigrade for 2 hours. Once the solution is obtained, electrospinning is carried out. The results show that the obtained fibers have an average diameter of 220 nanometers. It is possible to introduce bioglass particles with diameters on the order of 10 nanometers. According to mechanism explaining particle growth by the salt gel method, the bioglass particles introduced into the fibers are considered primary particles. In these particles, calcium 2+ ions are present in small proportions, with particle having a silicon monoxide calcium oxide composition being predominant.
Using bioglass particles obtained via salt-gel process in an acidic medium, which form agglomerates reaching a diameter of approximately 695 nanometers, makes handling difficult for producing hybrid organic-inorganic scaffolds using electrospinning, since the fibers obtained by this technique have diameters of barely 200 nanometers. Analyzing the surface of the bioglasses obtained by the salt-gel process under both acid and base catalysis using imaging softwares based on the light and dark contrast effects on the surface roughness, it can be stated that the acid-catalyzed sample exhibits greater surface porosity compared to the base-catalyzed sample. That is, it presents a greater absence of matter on the surface compared to that obtained by the basic method. In summary, using bioglass particles obtained by the casting and subsequent sieving is ruled out. For particles to be introduced in said polymer nanofiber matrices, it is recommended to use particles obtained by the salt-gel process via either acid catalysis or base catalysis. However, in the first route, the average agglomerate particle says on the order of 600 nanometers, whereas in the second route, basic catalysis particles have a diameter of 100 nanometers. Therefore, it's recommended to use particles obtained via the Stover method, and it is even advised not to resort to an in situ hybrid synthesis. The bioglass nanospheres obtained by the basic catalysis exhibit intraparticle and interparticle nanoporosity, as well as metal ions such as calcium 2+ and magnesium 2+, that induce an amorphous structure in the bioglasses. The low surface roughness suggests that wetting is governed solely by the chemical composition of the bioglass. Furthermore, the nanoporosity of the particles promotes capillary diffusion of the molecules such as water and low molecular weight alcohols. Notably, interparticle porosity is also present and is more pronounced in films composed of the acid-catalyzed bioglass. The presence of calcium 2+ ions in the structure further increases this porosity. These are the reference what we used. Thank you very much.